Scientists around the world are constantly looking for new methods to keep up and respond to a rapidly moving COVID-19 pandemic and a constantly changing novel SARS-CoV-2 virus. Vaccines have been a huge success, but their global rollout has been slow and non-equitable. To combat this, CanSino Biologics in China have developed an inhaled version of their COVID-19 vaccine, which has had early success in clinical trials. 2 inhaled doses of the vaccine, given 28 days apart, produced a similar level of protection measured by neutralizing antibodies to those of a single injection. And the lower dosage used means that production capacity can be boosted. It's hoped that technology like this will overcome some of the logistical challenges of a global vaccine rollout, getting more people protected more rapidly. But to keep ahead of the virus, we need more than just vaccines. A huge global collaboration has, at remarkable speed, broken down the complete life cycle of the novel SARS-CoV-2 virus. By understanding exactly how the virus enters, takes over, and exits human cells, it's hoped that new methods of prevention and treatment can be found. It also helps us understand what may lie ahead in the fight against future variants. But first, let's start with the basics. Here's a SARS-CoV-2 virus particle with its internal RNA genome and external membrane and spike proteins. When inhaled, the virus needs to reach an ACE2 receptor, found on the outside of cells in the throat and lungs. Connection is made via the spike protein, which we can look at in more detail. The bottom of the spike protein, or stalk, has hinges or joints allowing for fluid movement. It's thought this may help the virus locate and bind with an ACE2 receptor. This is unlike other viruses such as influenza, which have much more rigid external proteins. The bottom section here is the S2 subunit, and the top is the S1 subunit. This contains the receptor binding domain, the point where the spike physically connects to ACE2. Notable variants, including alpha and delta, have multiple changes in this S1 subunit structure, which studies suggest help the spike protein bind better to the ACE2 receptor. This, in turn, increases the transmissibility of the virus. Whether further changes in this S1 subunit structure could improve the binding of future variants and therefore increase transmissibility is still unknown, but scientists are certainly not ruling it out. The spike also plays a vital role in triggering our immune response or protection against SARS-CoV-2. After infection, or better still vaccination, the structure of the spike is remembered by the immune system. If detected again, the body quickly mobilizes antibodies to eradicate the virus, preventing infection or serious illness. Changes to the structure of the spike, like with the alpha, beta, delta, and gamma variants, makes it harder for the body to detect the virus and therefore mount a defense against it. New research has also found that SARS-CoV-2 uses glycans, which are sugar molecules, around the surface of the spike protein to help it hide from the immune system. Targeting the sugar coating is a potential avenue for new drugs and treatments. After the spike binds to ACE2, an enzyme called TMPRSS2 cuts the spike at the S2 subunit, facilitating rapid entry of the RNA genome into the human cell. This TMPRSS2 pathway is believed to be much faster than other viral entry pathways. For example, the original SARS-causing SARS-CoV virus from 2003 uses a pathway called endosomal entry, which is thought to be much slower and therefore less transmissible. Targeting the entry pathway is another area for drug treatments. The malaria drug chloroquine, which looked hopeful in lab studies for treating COVID-19 disease, disrupts endosomal entry. We now know the reason it did not work in real-world clinical trials for COVID-19 is because of the use of the TMPRSS2 pathway by SARS-CoV-2. After the viral genome enters the human cell, it hijacks it. A hijacked cell starts producing new SARS-CoV-2 virus particles, which eventually exit the cell, allowing SARS-CoV-2 to infect other cells in the body. Here again, the virus has another unusual trick up its sleeve. On the spike protein, between the S1 and S2 subunits, a SNP is made. This site, called the furin cleavage site, has been widely discussed since its discovery at the beginning of the pandemic. It's a trait not seen in the original SARS-CoV virus, but has been found in other viruses, like the highly pathogenic avian influenza virus. The furin cleavage site is used by the TMPRSS2 pathway from earlier. It primes the spike to be cut quickly by the TMPRSS2 enzyme, allowing rapid entry of the virus genome into the human cell. Studies have found that in the original SARS-CoV-2 virus, around 50% of the spike proteins were primed with this cut. The Alpha variant is believed to have more than 50%, whilst the Delta variant is thought to have more than 75% of its spikes primed with this cut. This is another explanation for the increased transmissibility of the Alpha variant, and then again for the Delta variant. It also highlights there is still capacity for a new variant to reach up to 100%. As cases continue to rise in this Delta variant wave of the pandemic, there are more opportunities for the virus to mutate and, under evolutionary pressure, become more transmissible and more immune evasive. Vaccines are a great success, but only one tool in the fight against the virus. Understanding exactly how it functions in the body enables scientists to explore other tools, such as targeting the sugar coating on the spike protein or entry pathways with new drugs and treatments.